Okay, guys, today we're going to have lesson 7.2 dealing with nervous system structure and function. Now, the first thing that we need to talk about is actually some cells that help support the nervous system. Now, collectively, we call these the neuroglia within the central nervous system, and they help to support, insulate, and protect the neurons that we find there. So we're going to start with astrocytes. Now, astrocytes are the most abundant. They are star-shaped cells. They will help hold everything together, and they also help form the blood-brain barrier between the capillaries that are found within the brain and the neurons themselves. So we see right here we have this centralized cell body, then all these projections coming out surrounding the capillaries, holding on to the neurons that we see in the background. Now, the next that we have are microglia. Microglia, these are spider-like phagocytes. They will help dispose of debris within the cell. So if something does get into the brain, maybe even damage neurons themselves, these microglia will help to dispose of that and get it out of the central nervous system. So we see, again, we have this centralized cell body, these projections coming out, branching into smaller and smaller pieces to phagocytize anything that may be that's in need of being removed. Now, next we have our ependymal cells. Now, ependymal cells will line the cavities of the brain and spinal cord. They are ciliated, and their purpose is to circulate the cerebrospinal fluid. Okay, so we can see right here these cells, they have these projections coming out, these cilia. They will move back and forth, back and forth, back and forth, and will help, they will help to circulate that CSF. Now, next we have our oligodendrocytes. The oligodendrocytes will wrap around nerve fibers in the central nervous system and produce myelin sheaths. Now, we'll talk in more detail about myelin later. Okay, but you can see right here we have this continuous wrapping, so to say, around the nerve fibers, and then we'll have an oligodendrocyte being located between many of them with what's called a process connecting the cell body to the individual neurons. Now, we also have satellite cells, which will help protect neuron cell bodies, and Schwann cells that are found in the peripheral nervous system. They form a myelin sheath there. However, they have a little bit different physiology than the oligodendrocytes do. So you can see right here with our Schwann cells, you have individual cells and then these spaces in between that are called the nodes of Ranvier. We'll talk about those in a second. Now, you can also see the satellite cells located around the cell body right here to give it a little bit of extra protection. Now, let's talk about what a neuron actually is. When we say neuron, we are referring to a nerve cell. So these cells are specialized to transmit messages. Some major regions of neurons are going to be the cell body and the process. So within the cell body, we have the nucleus. This is also the metabolic center of the cell. And then we have our processes. These are fibers that extend away from the cell body, and they travel to either the dendrites or the axons. So within the cell body, we have what's called the Nissl substance. So this is a very specialized endoplasmic reticulum. It's highly specialized in making our neurotransmitters, which will help transmit messages from one neuron to another. We also have the neurofibrils. This is an intermediate cytoskeleton. It's very important that neurons maintain their shape. Because we are transmitting these messages from one cell to another. We don't want to have it get folded, so to say, and cause that message to not be sent efficiently. So figure 7.4 gives this generalized model of what what a neuron will look like. So we have the cell body right here. You can see mitochondria labeled. You can see the neurofibrils within here, the Nissl substance, maintaining this centralized cell body with these projections, these dendrites coming off. These dendrites will carry messages into the cell body and then transmit it into an axon through the axon hillock. And then we can either, have a single axon. So we'll just ignore this part right here. For a second, you have a single axon with multiple axonal terminals down here, or sometimes we can branch into we can have multiple branches called collateral branches. So in figure 7.4b right here, you can kind of count with me. It looks to be 1, 2, 3, 4, 5, 6, maybe 7, 8 different projections coming off these dendrites that you can see coming off from the cell body. So again, the cell body is where the nucleus is. There's a very large nucleolus there. Again, that's done to build the centralized region for ribosome production to make all of these neurotransmitters on the Nissl substance. Now, the process outside the cell body, we classify them as two ways, either dendrites, which will conduct impulses toward the cell body, or axons, which will conduct them away from the cell body. So to jump back to figure 7.4, we can see our dendrites right here, and we've even added arrows to show the general motion of which those messages are transmitted. So these axons end in axonal terminals. The terminals contain vesicles with neurotransmitters. We'll talk about those when we get to nerve physiology in a couple of minutes. Now, what's important to note is that our axon terminals are separated from the next neuron by a gap called the synaptic cleft. So this occurs when we have a gap between adjacent neurons, and the synapse is going to be the junction between nerves. Okay, so we're not going to have a physical connection between an axon and a dendrite. So we have our axons right here. Let's imagine there's another nerve right here. They aren't going to physically be connected. So again, we have this myelin sheath that's produced by our Schwann cells. It's a whitish fatty material. It helps speed up nerve impulses. And the nodes of Ranvier, those gaps that I talked about. Again, I'll jump back to theory 7.4. So the most commonly accepted theory is that we have these so that there's more flexibility of our nerves. Remember, our limbs are moving around. So these nerves are being stretched and compressed. Those nodes of Ranvier allow for us to still have that insulation, but for it to not, be crushed or torn apart through movement. Okay, so figure 7.5 outlines how myelin will begin producing. So you can see we have our Schwann cell nucleus right here, and it will begin producing in a circular motion. And what's important to note here is this, that it actually will tuck inside. Okay, so the nucleus of the Schwann cell is outside, and we will wrap layer after layer after layer after layer after layer. And the outermost layer we'll call the neurolemma, and then this myelin sheath is found within there. Now, let's talk about some location of some structures at this point. So most neuron cell bodies are found in the central nervous system. So if we have ones that within the central nervous system of the gray matter, we find cell bodies and unmyelinated fibers. Okay, so within the brain, parts of the spinal cord, and then we have nuclei, which are going to be clusters of cell bodies within the white matter of the central nervous system. These will aid in communication with the peripheral nervous system. Now, in terms of outside of the central nervous system, we can have what are called ganglia. Now, ganglia are going to be collections of cell bodies outside the central nervous system, and they really help coordinate the function of large parts of the body. So we have ganglia for the appendages to help coordinate their movement more efficiently. Now, in terms of the functional classification of neurons, we have sensory neurons and motor neurons. Now, these afferent or sensory neurons, they always carry impulses from a sensory receptor to the central nervous system. We have our cutaneous sense organs and our proprioceptors, which will detect things like stretch or tension. And then our efferent or motor neurons, these are going to carry impulses from the central nervous system to either the viscera, the muscles, or glands. Okay, so smooth muscle as well. The viscera are digestive tract glands to release hormones or to regulate tons of stuff within the body. Now, there's a very interesting passage in your textbook that outlines all of these. I do recommend you read that. And it's very interesting how all of these work. This diagram has always kind of concerned me. If you look at figure 7.7a, I don't know if it's just the angle, but this guy has like really cut into his finger. Okay, but you can see that it outlines this deep pain receptor right here. I mean, he would have like clean cut all of that that you could see. Now, you can see with our stretch and tension right here, it's like when you're lifting weights, this feeling of pressure that you have is what's demonstrated here. Okay, now lastly, we have our interneurons or associated neurons. Sometimes we have a neuron that serves as kind of like a go-between or a messenger, so to say. So this is found in neural pathways in the central nervous system, and they will directly connect a sensory and motor neuron like we see in a reflex arc. So figure 7.6 gives the generalized diagram of a reflex arc where we have a afferent nerve right here, our dendrite, afferent nerve, interneuron, motor neuron, and effector. Now, this is actually a question in your assignment today. Okay, so this is the 5 minimal components that you need for a reflex arc. You need a receptor, an afferent nerve, an interneuron, a motor neuron, and an effector. Okay, so our structural classification of neurons continued. We have multipolar neurons. You have many extensions from the cell body. You have all these dendrites projecting out. Typically, you'll have a single axon hillock right here, and you could branch into multiple or collateral axons ending in axonal terminals. You can see right here. And this is a good time to bring up. Generally speaking, good way to distinguish between dendrites and axonal terminals. Dendrites typically are going to be more pointed, whereas axonal terminals are going to be more rounded. We'll talk about why that is in just a second when we get to nervous physiology. So bipolar neurons, you can see right here in this example, the cell body is embedded within a single long pathway. You have your dendrites right here, axonal terminal over here, and the cell body located roughly equidistant between the two. Now you have the unipolar neuron. So in this case, you have a short signal process where the cell body is outside of the main pathway. Again, equidistance. The peripheral process is always going to be on the dendrite end. It's going to be on the efferent end. And afferent end, and then your central process will be your axonal terminals on the efferent end. Now, your functional properties of neurons, so they have to be irritable and conductive. So when we say irritable, they have to be able to respond to a stimulus. So when we say respond, we mean sense that there's been a change. So if you have some like superficial nerve damage, you could have damage to the dendrites that are in some of those proprioceptors that we talked about a few minutes ago. Now, the other thing is they have to be conductive. So when your foot or your hand goes to sleep, so to say, what's actually happening is you have blocked that conductive pathway of your nerves, causing it to not be able to make it to the integration center, whether it is an interneuron, ganglia, or even the central nervous system. So when you stand up, so to say, and your foot is asleep, that burning sensation is actually the stimulated nerves finally getting that message to the brain, and it has to restore itself. Now, before we move into nerve impulses, getting into nerve physiology, I know this is a long lesson, so if you want to take a second and take a break, this would be a good time to do so. Get up, stretch, do whatever it is you need to do. Okay, so we're going to pick up with nerve impulses now. Now, we need to create this idea of what we call a resting neuron. So, at rest, the plasma membrane of a cell is polarized. Now, what that means is that we have a difference in charge. Now, in this example, we have fewer positive ions inside the cell than outside the cell. So what that means is that inside the neuron, we have a negative charge. Outside of the cell, we have a positive charge. Now, when we depolarize a cell, we simply flip this. So a stimulus will depolarize the neuron's membrane, causing it to change. So the exchange of ions initiates what we call an action potential in the neuron. So let's talk about how we actually do that. Now, again, with this resting membrane electrical conditions, you can read through all of this. What we see, we have sodium, potassium here. We have more sodium outside the cell than in, the same for potassium. Now, the membrane is relatively impermeable to both, okay? So what we see, negative charge here, negative charge here. So a stimulus will initiate a local depolarization. Now, in terms of local, I want you guys to imagine for a second that we're talking just right here. That's it, just to the left of where the cursor is. All this over here, we're not going to work on it right now. Okay, so it will, the stimulus changes the permeability of a patch or this small part of the membrane. So sodium will move in and into the cell, and it's going to cause a very rapid change at that point. So it's going to become more negative outside the cell. And at that point, the action potential begins. So if the action potential starts, it's propagated over the entire axon. Now, guys, this is a true all or nothing pathway. That's a very important thing to remember. Okay, it is all or nothing. So impulses will travel faster when fibers have a myelin sheath. So with the oligodendrocytes in the central nervous system, the Schwann cells in the peripheral nervous system, it's important that you remember that. Don't forget that part, okay? Now, it's at this point, once this depolarization occurs, it's going to transition through the entire neuron, okay? So we got our dendrite over here. Stimulate that. It's going to travel through the cell body and then down our axon, okay, propagates through the entire axon, and the action potential propagates rapidly through the entire length of the membrane. Now, once that happens, though, we got to get back to how we started. So this is a process called repolarization. Now, potassium ions will rush out of the neuron after sodium ions rush in, which will repolarize the membrane. So the sodium-potassium pump is what's called a symporter. It'll use ATP and will help to restore the original configuration. Okay, so you can see right here, this action potential is gone on, it's done its thing, and repolarization begins. So when I said symporter, okay, it brings 2 potassiums back in and sends 3 sodiums out at the use of some ATP. So this will get us back to our original charge, and it's simply just going to wait there for the next message. Okay, so impulses are able to cross the synapse to another nerve. So again, those neurotransmitters we talked about, they're released from the axon terminal, and the dendrite of the next neuron has receptors that hopefully are stimulated by the neurotransmitter. Now, they do not always, they are not always received. Sometimes they are not. We'll talk about that later in this chapter. So an action potential is started in the dendrite of the next one, or if it is at the effector, it causes some type of physiological change. Okay, so figure 7.10 outlines this. So let's start with this right here. We've got this axonal terminal at a dendrite. Okay, and then right here we have the synapse. So let's take a close-up look at the synaptic cleft. So we have had this action potential, all that happened in step 1. Now, we have our vesicles. This is our neurotransmitter, acetylcholine right here, travels to the membrane. Now, once it does, it's going to fuse. And once it fuses, it's going to open. It's going to release this neurotransmitter into the synaptic cleft. And you can see we've got a whole bunch of them. We'll say like 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, we'll say 15. But only 2 have made it to the neurotransmitter. Okay, now, if we zoom back out for a second, it's important to remember with the synaptic cleft, it's open to the outside environment, okay, to the extracellular environment. So there's no guarantee that those neurotransmitters, all of them, will bind to the receptor. And there's a lot of medicinal therapy that focuses on getting neurotransmitters to the right place. All right, so lastly, let's talk about reflex arcs for a second. Now, with a reflex, this is rapid, predictable, and involuntary. It occurs over pathways called a reflex arc, and it's a direct route from a sensory neuron to an interneuron to an effector. So let's go through this step by step. Now, you have this stimulus at the distal end of a neuron. It's going to trigger we'll say a cutaneous sense organ. We see right here this kind of light blue substance. That's going to send a message through our afferent neuron, our sensory neuron, to the gray matter of the spinal cord, where we have an interneuron, in this case representing the integration center. And then that's going to send a message to our motor neuron, sending a message to our effector, in this case being skeletal muscle. Now, what is really interesting about this, about reflex arcs, is that if they are pain associated, that your body will react before you feel the pain. Okay, so this one shows the pinprick in in your finger, or your hand on a hot stove example. So your body will react before you actually will sense that pain. It takes the brain longer to comprehend pain than it does for you to have a response to that. Okay, so with our somatic reflexes, you have things like activation of skeletal muscles, the one that we just walked through. But with the autonomic reflexes, these are the ones I want to talk about for a minute. So you have things like smooth muscle regulation, heart rate and blood pressure regulation, regulation of glands within your body, and also digestive system regulation. So this is all going to be done on its own, but it's important to note that other substances like caffeine and alcohol, they can affect these. They can affect these autonomic reflexes. Okay, so caffeine is a stimulant. Alcohol is a depressant. So with a stimulant, it's going to make you more heightened, whereas alcohol being a depressant will make you a little more delayed, so to say. If you drink a lot of caffeine, you can expect your digestive system to slow down, your heart rate and blood pressure to increase. You'll see it act as a diuretic, causing increased activity of the urinary system. All that's going to be reflexes in response to some type of chemical change. Okay, and of course the classic knee-jerk example is an example of a 2-neuron reflex arc. Now, I've had students every year in the past ask, when you go to the doctor as an adult, why don't they do the knee jerk check anymore? Well, it's used as a way to assess proper nerve development. Now, if you go to the doctor as a child and you're going through your checkup and all of that, and they test your knee and they see that they tap your knee and you don't have that reaction, that it can be indicative of some other developmental issues that may need to be examined further. Okay, so if something as physiologically basic as a reflex arc is not functioning properly, that they need to do further examination to see if there are other deficits that may be present. So this wraps up our lesson. If you have any questions, put it in the comments below. Email me at mr.tucker.schs@gmail.com.